It's my pleasure to present to you the results of the primary analysis of the ZUMA-1 pivotal phase 2 trial of Axi-cel in patients with refractory aggressive B-cell non-Hodgkin lymphoma. Here are my disclosures. So I think there is a significant unmet need in patients with refractory aggressive B-cell non-Hodgkin lymphoma. In a recent meta-analysis in the SCHOLAR-1 study with over 600 patients, we observed that the best overall response rate with existing therapies is 26%, and the median survival is only about 6.5 months. So we tested Axi-cel in this refractory patient population. Axi-cel is an autologous chimeric antigen receptor T-cell therapy with a CD3 zeta CD28 based signaling that recognizes and eliminates CD19 expressing cells. In the phase 2 portion of the ZUMA-1 study, there were two cohorts. Cohort 1 included patients with diffuse large B-cell lymphoma, and cohort 2 included patients with primary metastatic B-cell lymphoma and transformed follicular lymphoma. To be eligible, the patients had to have chemotherapy refractory disease that was defined as no response to last chemotherapy or relapse within 12 months after autologous transplantation. The primary endpoint for this phase 2 portion of the study was objective response rate in the first 92 patients that were dosed on the study. Key secondary endpoints included duration of response, overall survival, safety, as well as CAR T levels and cytokine levels measured in the peripheral blood. So we enrolled a total of 111 patients across 22 different sites. The product could be manufactured in 110 of 111 patients for a 99% manufacturing success rate. The average turnaround time from apheresis to delivery to clinical site was 17 days. Following the manufacturing of the product, the patients received conditioning chemotherapy with cyclophosphamide at a dose of 500 milligram per meter square and fludarabine at a dose of 30 milligram per meter square daily for 3 days. And 2 days later, they received Axi-cel at a target dose of 2 million CAR-positive cells per kilogram body weight. Altogether, 101 patients were treated with Axi-cel. 10 patients did not receive the product, either because of serious adverse events in 7 patients, not having measurable disease in 2 patients, or the product being unavailable in 1 patient. This study did not allow bridging therapy following leukapheresis and enrollment. As I mentioned, the primary analysis is based on the first 92 patients that were dosed, but at the time the 92nd patient was dosed, there were 9 additional patients that were enrolled and leukapheresed in the study. So these 9 were included in the modified intent to treat analysis for a total patient population of 101. The data cutoff for this presentation is January 27, 2017, and the median follow-up was 8.7 months. Here are the patient characteristics. The median age of the patients was 58. Almost a quarter of the patients were 65 years or older. 85% of the patients had disease stage 3 or 4. Almost half the patients had an IPI risk score of 3 to 4. Close to 70% of the patients had 3 or more prior therapies, and more than 50% of the patients were refractory to 2 consecutive lines of therapy prior to enrollment. In terms of the refractory subgroups, 77% of the patients were refractory to second or later lines of therapy, and 21% relapsed post-auto transplant. The study met its primary endpoint for the overall response rate with a p-value of less than 0.0001. In the combined group, the best overall response rate was 82%, and the CR rate was 54%. In cohort 1, the overall response rate and the complete response rates were 82% and 49%, and in cohort 2, they were 83% and 71% respectively. The responses were consistent across key covariates, including the refractory subgroups, the age groups of the patients, the disease stage, the IPI risk score, and whether the patients received tocilizumab or corticosteroids for management of adverse events. The responses appear to be quite durable, with 44% of the responses ongoing at a median follow-up of 8.7 months, and 39% of the patients remain in complete remission. In cohort 1, 36% of the responses are ongoing, with 31% being complete responses, and in cohort 2, they were 67% and 63% respectively. And here are the Kaplan-Meier curves for the duration of responses. At a median follow-up of 8.7 months, the median duration of response among all responders was 8.2 months, shown here in the blue curve. The median duration of response for the complete responders has not been reached, and for partial responders is 1.9 months. The median PFS on the study was 5.9 months. To determine the impact of covariates on the durability of the response rates, we looked at the 6-month PFS rates, and similar to responses, we did not see any significant differences in the 6-month PFS rates in the various subgroups, including the refractory subgroups, the age groups, disease stage, IPR risk score, and whether the patients received tocilizumab or corticosteroids. The median overall survival has not been reached at a median follow-up of 8.7 months, and as compared to the historical data of the SCHOLAR-1 study, the 6-month overall survival rate of 80% in the ZUMA-1 study was markedly superior to the 55% 6-month overall survival rate observed on the SCHOLAR-1 historical data. And here is a representative case. This was a 62-year-old male that was previously refractory to 4 lines of therapy for his diffuse large B-cell lymphoma. At the time of enrollment, he had multi-compartmental bulky lymphadenopathy as well as large lung masses. He achieved a complete remission with Axi-cel and remains in complete remission now 12 months later. And here are the most frequent grade 3 or higher treatment-emergent adverse events. The majority were cytopenias, thought to be related to conditioning chemotherapy. Encephalopathy related to Axi-cel was observed in 21% of the patients. In terms of cytokine release syndrome and neurological events that were thought to be related to Axi-cel, these were generally reversible. Grade 3 or higher CRS was observed in 13% of the patients, and grade 3 or higher neurological events were observed in 28% of the patients. There were 3 grade 5 adverse events, one due to hemophagocytic lymphohistiocytosis, or HLH, and a second due to cardiac arrest in the setting of CRS. A third patient died of pulmonary embolism that was thought to be unrelated to Axi-cel. All neurological events resolved except for one subject that has ongoing grade 1 memory impairment. Altogether, 43% of the patients received tocilizumab and 27% received corticosteroids for management of these adverse events. There were no new Axi-cel related grade 5 adverse events between the interim analysis and the primary analysis of the study. Moreover, it appeared that the grade 3 or higher CRS and neuro events appeared to decrease over time during the course of the study as we refined the management guidelines for the toxicities and the sites gained more experience in the management of these adverse events. There was significant heterogeneity in the starting apheresis material when we looked at the percent T cells in these products, but despite this, Axi-cel could be manufactured in all but one case for a 99% manufacturing success rate. Looking at the phenotype of these CAR T-cell products prior to infusion, based on CCR7 expression and CD45RA expression, we observed that the median naive T-cell population was 14% in the 101 patients, and the central memory T-cell subset was 27%. The two subsets that are known to be associated with in vivo T-cell proliferation and long-term persistence. The median CD4-CD8 ratio for the 101 patient was 0.9, and when we grouped these patients into 4 different quartiles based on the CD4 and CD8 ratio of the Axi-cel product, we did not see any significant differences between the overall response rates, the complete response rates, or the development of grade 3 or higher neurological events or CRS. We measured CAR T-cell levels in the peripheral blood of these patients by quantitative PCR, and we observed CAR T-cell levels peaked within 7 to 14 days and declined over time, but they were persistent at 6 months, the last time point that has been assessed so far on the study. The expansion of the CAR T-cells was associated with overall response rate and grade 3 or higher neurological events, but not grade 3 or higher CRS. The CAR AUC levels, defined as the cumulative levels of CAR cells per microliter of blood over the first 28 days post Axi-cel infusion, was 5.5-fold higher among responders compared to non-responders. In addition, the CAR AUC levels were about 2.5-fold higher in the group of patients that had grade 3 or higher neurological events versus those who did not. So to summarize, Axi-cel significantly improved overall response rate with 44% of the responses ongoing, median follow-up of 8.7 months. The study met its primary endpoint with a p-value of less than 0.0001. The CR rate of 54% is 7-fold higher than the historical control rate of 8% observed on the SCHOLAR-1 study, and the median overall survival has not been reached to date. Axi-cel could be administered with a manageable safety profile. The CRS and neurological events were generally reversible, and the rates appear to decrease over the course of the study as we refined the management algorithms for these toxicities. The use of tocilizumab and corticosteroids did not negatively impact response. The expansion of the anti-CD19 CAR T-cells was associated with overall response rate and grade 3 or higher neurological events. The Axi-cel could be manufactured in 99% of the patients, even from this heavily pretreated patient population with a 17-day turnaround time, which I think is critical for these patients that have aggressive lymphomas. So this is the first pivotal trial of anti-CD19 CAR T-cells in refractory aggressive non-Hodgkin lymphoma that demonstrated significant clinical benefit for patients lacking curative options. So with that, I'd like to end by acknowledging the patients and the families that participated in the study staff at the various institutions, as well as support from Kite Pharma and the Leukemia and Lymphoma Society. Thank you for your attention.
Okay, I think we have time for a couple of questions. Maybe I could start by asking you, do you see this eventually perhaps replacing autologous transplant in lymphoma?
Yeah, that's a great question. So I think that is definitely on our minds, because even in this third line and beyond, we are seeing that more than 40% of the patients have ongoing responses. So I think the next step obviously is to test it in the second line setting, head-to-head with transplant, and those studies are being planned currently.
Okay, next.
Yeah, hi. Very nice, very interesting study. Congratulations on this very large cohort. You showed phenotyping data on the apheresis, where you identified the various subsets. Do you know if there's any association between those subset distributions and response to therapy? And second, have you looked at the infusion product, and did you see any phenotypes that stood out? And third, have you looked at exhaustion, which may play a bigger role in non-Hodgkin's than others? Did you see any correlations there with response to therapy?
Yeah, so to date, we have not seen a correlation between the subsets that I've shown you in terms of the naive central memory and effector memory and effector cells versus response. We are looking at additional subsets, functional subsets of this Axi-cel product to see if there are correlation between certain functional subsets versus response. Those studies are ongoing and will be presented at a future meeting.
One last question. Sorry.
And in terms of the exhaustion, we have not characterized that yet.
One last question.
Can you discuss the CRS death?
Yeah, so this was a patient that had grade 4 CRS and was being observed in the ICU. And this patient, while being observed in the ICU, all of a sudden developed cardiac arrest. And despite CPR and other interventions, the patient did not recover.
Thank you very much.